pleasure today to introduce Stephanie, who's going to be giving her dissertation defense talk today. These events always bring back old memories, and my oldest memories of Stephanie are when she first came to us from the University of Texas at Arlington. And all of her undergraduate experiences were in mathematical biology, and so my head was thinking that we were going to do some modeling project together, and it didn't take long. Well, it did take a while. It took me about a year to realize that really wasn't what she was about, and that I think what I mistook for that interested in mathematical biology, it was really more of an insight to be able to identify really interesting questions, and courage, the courage to follow it using whatever technique was called for by the question at hand. And so she took us down a completely different path, following the lizards, I guess, down this completely different path into biochemistry, forging this relationship with Helena Soini and Milos Novotny in the Institute for Pheromone Research. Yeah, and along the way she's been really very successful in lots of ways, lots of awards, lots of travel awards, lots of she was in the CTRD, got a CTRD fellowship, and then spent a year getting, I forgot the name of this current fellowship that you're on, a dissertation year.
President's Diversity.
The President's Diversity Fellowship for this final year that was very helpful since I moved. I think the most, I'm going to let her tell you about her research, but the thing that drives me the most, that excites me the most about it, is really the combination or the balance between individual insights and complexity. So she has some situations that you'll see where she'll be talking about individual compounds that are really important in terms of signaling, and then other cases where it looks like the combination of compounds is really what's driving the story. And I think it's that balance between the two and the going back and forth that maybe is the power of underlying what she's been doing. So I'm going to let her tell you about her research now. Communication breakdown, evolution of territorial chemical signaling in a diverse lizard genus. Stephanie Campos.
Thank you for that introduction. Before I begin, I want to acknowledge my main advisor, Amelia Martins, who has helped me form these projects and really has been instrumental and an excellent mentor for me through the last 6 years of this. Being able to detect, interpret, and respond to signals within an animal's environment is critical to their fitness. We can learn a lot from an animal by studying their communication. We can determine the developmental history of a bird based on the vocal repertoire of their songs. We can determine the social relationships of weakly electric fish by examining the changes in EOD discharge frequencies. We can examine heart rate of vole pups by looking at their ultrasonic vocalizations, and the specific series of push-up displays in Scoloporus lizards will tell us what species they are. So alongside all of these really specific forms of communication in these animals, all of them communicate with chemical signals. Whether it's internally regulating physiological processes of the endocrine system, or whether it's emitting signals to the external environment used to communicate across conspecifics. How an animal responds to these odor signals is really important to their survival. Social bees emit alarm pheromones in areas where a recent predation attempt has occurred, and by detecting those, they know to avoid that area. Bombyx mori silk moths use sex pheromones to find potential mates. Scent marking is a really specific type of chemical signaling that is often complex. We associate it frequently with territoriality, and we often, or historically at least, it's been viewed as a method for competitor deterrence. So one of the driving questions in my research is how are chemical cues shaped along with social dynamics of an animal? Natural and sexual selection might shape chemical signals to be conspicuous within the natural sense already in the environment that an animal lives in. They also can be shaped by environmental extremes of the environment. For example, we know that scent marks are affected by habitat temperature and moisture levels. Generally speaking, really hot environments will volatilize signals more quickly and decrease the total lifetime of a signal. For example, these two jaguars live in very different environments. One lives in a really tropical habitat where there's a lot of dense vegetation that produces their own compounds. So if it's adapted to that type of environment, it might have very different chemical signals to communicate with conspecifics versus the jaguar that lives in a very montane, dry habitat where there's very little vegetation to overcome those smells. But because these animals share a phylogenetic history, they might be constrained by the same physiological constraints, and which would impact the types of compounds that they can produce. So aside from these really broad views of environment, there are also microenvironmental impacts on the social interactions of animals as well as their signals. Chemical signals might simply be the byproducts of their metabolism, constrained by different parts of their physiology. For example, in ring-tailed lemurs, they can tell the difference between the scent from a healthy individual versus the scent from an immunocompromised individual. We also know that animals are covered in microbes. They impact the signaler's behavior through physiology, metabolism, immune function, and mood, but they can also have an indirect impact on the social signals of a host, and in turn influencing the behavior of the receiver. Okay, so to address a lot of these broad themes, I used the territorial lizards, Scaloporus. These are really small lizards in a really big genus. They are behaviorally, phenotypically, geographically diverse lizards, and they're named for the specialized row of femoral pores on their inner thighs, being highlighted here on this territorial male. And if you take a closer look, in the breeding season, males will enlarge these femoral pores, and they amp up the production of the secretions, along with, as testosterone amps up, these secretions also amp up. The females have these pores, but they don't produce them in the same way as the males do, the secretions, that is. Beneath each pore is a gland, a femoral gland, that produces these secretions, and they're holocrine in nature, meaning that as new cells form, the old cells are pushed inward and they migrate up and out of the pore, and as they do so, the cells disintegrate and release the contents that were in the cell. So that's what actually the secretions are made of. These chemical signals alone have been shown to elicit aggressive responses in males, and we'll go into a little bit more about the sex differences in this genus. So on the left, you see a picture of a female on the right and a male on the left of the same species. Frequently in Scaloporus, we have sex differences in morphology and coloration, which can differ depending on the species. But you can see here that the female has a much more rotund belly. The male is much more colorful. And this male on the right is in an aggressive stance. He's alert, pushed up, and he's showing off that aggressive blue coloration that's on his ventral side. Typically, when you see males in the breeding season, this is what they look like. They're perched in a really conspicuous area, usually a big rock or a log in their territory, and they'll visit these landmarks, patrolling their territory throughout the activity period. They'll visit other females, and they will fight off other males. This picture on the bottom shows you a typical male-on-male interaction in the breeding season. The one on the left is lunging at and biting the one on the right. The one on the right is in an aggressive stance like this one here. The females, on the other hand, tend to stick in a really small area of their territory. They are territorial, but they don't seek out interactions the way that males do. Okay, so now that I've introduced you a bit to the themes and my system, I think what the driving questions of my research, the main theme, is this, that I'm trying to break down the different parts of communication by studying the evolution of chemical communication to ask about the behavioral significance and the different components in the signals and how they're shaped over time with evolution and with the behavioral interactions of a species. So in part one, I'll really target the behavior, looking specifically at the effects on territorial space use. In part 2, I'll look at microenvironment, specifically the microbes that are associated with these femoral pores. In part 3, I ask about the evolution of chemical composition, looking at 12 different species of Scoloporus, and ask about the relative roles of habitat climate, as well as behavior. And in part 4, I ask about the individual variation within a species and whether we can tell anything about the phenotype of an individual based on their chemical signals. So in part 1, I ask, do intruder odors impact territorial space use? This diagram here gives you an idea of the general idea of the project. So on the right, you see a resident male, and he's perched on top of a landmark, a log in this case. The area surrounding him represents his territorial space. The male on the left in blue represents a simulated intruder male. And what we did was introduce the scent of a simulated intruder into the territory of a resident male. And then we measured his behavioral space response. So this work was done with the help of 2 undergraduates, Chloe Strauss and Christian King. They helped in the field as well as with the thoughts and the writing behind this project. This took place at Lake Monroe here in Indiana and used Scoloporus undulatus, which are eastern fence lizards. So briefly, we went around and collected territorial males. That means that they were conspicuous in their territory, perched on a log that looks much like this or on a really big rock. And we used a noose tethered to the end of a fishing pole to catch them. And we gave them unique identification marks that look a lot like the one on the lower left. So this particular male has a red dot over a white dot. And that allowed us to see them from far away with binoculars and determine who they were. So we released these males back into their territorial home space, and we observed them for a time being. We identified territorial residents as well as the landmarks that they associated with based on how many times we saw them on a particular landmark. If we saw the same male on the same landmark at least 3 times, we considered it a resident male of that territory as well as that landmark. So what we were doing was introducing a treatment into this territorial male's space. We had chemical treatments which had the scent of an intruder male. We collected that male from a different area of our site so that to minimize the familiarity between the 2 individuals. For half of the males, we gave a control treatment which had no added scent. These Q papers were made up of black construction paper and were shaped like this X here. We left them for a period of 24 hours, and after that period, we came back and recorded behavior. We repeated this process 5 times per landmark. So each time we recorded behavior, we removed the old cue and put a new cue in the territory. If a male received a chemical cue first, they received a chemical cue for the entirety of the 5 different days. If they received control, it was consistent throughout those 5 days. Okay, so first we measured activity. This was a presence-absence measure, so if the male was around and active and conspicuous and behaving near the landmark, it was considered present. We looked at a 15-meter radius around the Q paper, and we also recorded the presence of any other females we saw, as well as any other males. Any male that we saw that was not the resident male, the focal individual, we considered him a non-resident male. And this is what the Q papers look like in the field. This is a zoomed in picture. We fit the edge of the paper into the bark, and so it would not be, it wouldn't mess with their locomotion. This is what we found. In this picture, you're looking at the response of resident males. On the left is the resident males that received a chemical treatment. On the right, control. And we're looking at the proportion of days that they were active in the dark bars, so when we saw them present. And we see that resident males were more likely to be active, or they were active on a higher proportion of days, when there was a chemical cue versus a control cue. And we saw the same effect for females, where they were active more frequently when there was a chemical cue. We did not see the same for non-resident males. They were equally likely to be active near either cue. So next, for every active lizard, we measured the distance between the cue paper and the lizard that we saw. And this will be given in meters on the y-axis. For resident males, we found that they perched closer to the chemical cue, but this wasn't a significant difference. And again, we find the same thing in females. But for non-resident males, while they also perched closer to the chemical cue, this was a significant difference. They were much farther, about 9 meters from the control cue versus 1 meter from the chemical cue. So to recap, we found the same results in resident males as we did in females. This could be due to territoriality in general. We know the females are territorial, so it could be for territorial defense for both resident males and females, or it could be for different reasons. The females, we know, don't seek out copulation, don't seek out reproductive opportunities. But this activity, this increasing activity when there's a new male around, might allow them to gain more reproductive opportunities. For non-resident males, while they weren't more active when there was a chemical cue, they were closer when there was a chemical cue. And so, regardless of the sex or resident status of the lizards, they seem to be attracted to these male cues. And if you look across all of them, they all perched about 1 meter away from the cue, regardless of their sex or resident status. So what does this mean for the field of spatial and chemical ecology? Well, territorial scent marks are historically thought to deter intruders, but we're showing that intruder scent marks are actually attractive to lizards, and they have a real impact on the territorial space use of these lizards in naturally formed territories in the field. I think we might be seeing some form of semi-gregariousness in these lizards. Territorial animals are often contrasted to really social animals, but we know that there are really social animals who are also territorial, and I think what we're seeing here is another form of territoriality, where they're actually attracted to at least the scent of other individuals. It could be that they're attracted to it just so they can fight, but they still have some benefit from being attracted to that scent. Finally, this study highlights that if we focus on the immediate measures of behavior in response to signals, this could overlook subtle effects that those signals have on behavior. And so going forward with animal communication studies, we should consider the fact that there may be some effect on behavior of a signal that is in a different time frame than the one we're measuring. Okay, part 2. Microbes have always been viewed as these sort of puppet masters of animal behavior, controlling how an animal behaves. We know that gut microbes alter the sex hormones of animals, specifically testosterone, and testosterone in turn alters the microbial communities of animals. Communities of microbes have been linked to different sexes, status, whether dominance or reproductive, in hyena clans. And in meerkats, it's been shown that until sexual maturity, meerkats have the exact same microbial communities in their scent glands. But once they hit that reproductive maturity and their hormones begin to differ, so too do their microbial communities in the scent glands. So there's this feedback interaction between hormone-mediated traits and the microbial communities associated with the host. Because we see these differences in secretion production in males and females, I wanted to know, are those sex differences reflected in the microbial composition of these communities at the scent-emitting pores? So we took a sample of the microbes from the femoral pores of both males and females in the field. This was done with the same species as before, but this time at Cedars of Lebanon State Park in Tennessee. We swabbed the femoral pores of 15 females and 9 males. This gives you an idea of what the differences look like in these femoral pores. So the females do have them. We brought these samples back to IU, and with the help of Irene Newton, we were able to break down these microbial cell walls, and then in the SysAb lab, we were able to extract the DNA using the Promega Maxwell. And with the help of CGB, we were able to do PCR amplification and create library sequences. Specifically, we sequenced the V4 region of 16S ribosomal DNA. And when we got our sequences back, we identified them through 3 different databases. All of these databases agreed to the phyla, which is the ones I'll show you here in a second, except for one particular taxa, which we classified as unknown. We calculated different community measures. These are really common ecology measures. The first is richness, which is basically the number of different taxa in a sample. The second is diversity measure, which accounts for both the richness as well as the proportion of a particular taxa within the community. We also calculated evenness, which is a score from 0 to 1, 1 being the most even, saying that all taxa are abundant in the same amount. And then we calculated the Bray-Curtis dissimilarity index, which again is a score of dissimilarity, 1 being the most dissimilar, which I'll go into here. So the Bray-Curtis dissimilarity index, 1 means that, that they share no taxa at all, and 0 meaning that they have the exact same composition. So in this, we have females in orange and males in teal. And you can see that the orange is interdispersed with the teal, suggesting that microbe communities are very similar between the sexes. They don't aggregate and separate like we would expect. So this showed us that there were no differences in microbial community composition, and there is our analysis of similarity score there. We also found no differences in any of the other measures of community, specifically richness, the number of taxa, evenness, and diversity, again suggesting that males and females look very similar in terms of their microbial community structure. The graph on the left shows you the main phyla that were in these lizards. So Proteobacteria was the most abundant, followed by Actinobacteria, and this is an average for both females and males. This graph on the right shows you the abundance of the top 15 taxa. These were all shared, so they were abundant in at least 1% of the total taxa, and we found no differences in any of these either. So despite what I thought would happen, we found that microbial community composition does not reflect sex differences in the production of secretions. If microbial communities are important in producing the chemical compounds that these lizards use, this might suggest that males and females produce very similar types of secretions. We've been unable to analyze the composition of females because they produce so little. It'd be really hard to get enough from lizards in the field to even look at what's in them. It's also possible that there are other physiological traits that better explain these composition that we're just not looking at here. So maybe it's more of health status or physiology rather than sex alone that explains microbial composition. Similar results have been found in juncos. Both males and females have very similar microbial communities, and the differences come with other types of social interactions. Speaking of social interactions, we know that in hyenas, the more similar the microbes, the closer in the clan two hyenas are. So hyenas that are within the same clan have very similar microbial communities, whereas if you compared them to a clan member of a different clan, they would look very different. And with social baboons, it's also been shown that the gut microbes that they possess look a lot more similar to the microbes of baboons that are in close social interactions with them than they do to other individuals that they don't encounter very frequently. So again, I think what we're seeing here is this effect of a semi-gregariousness in these animals. Often we see a connection between sociality and the types of microbes as demonstrated by these animals. And we know that lizards, as they move around their territories, rub their femoropores over substrates that are shared with other individuals. So it could explain why these microbes look so similar between the sexes within this population. Okay. In part 3, I discussed the evolution of chemical composition looking at 12 species. So the first aim of this study was to look at what's in the secretions. So we collected secretions from 12 different species. Here's a representative, 4 of them, in the field, and then we brought those secretions back to the Institute of Pheromone Research, where we chemically analyzed them using gas chromatography and mass spectrometry. You can see the secretions in these small vials here. It's very little. We did some chemistry and extracted volatiles from them using these stir bars. These stir bars are magnetic and they're covered with a special plastic, so when we mix this around with our secretions, the volatiles get embedded in this special plastic, and that stir bar is actually what we analyze, and you can see it here in this tube. And so after a program of heating and cooling cycles, we get back something that looks like a very messy version of this chromatogram. We can filter it for the peaks we're interested in, and then we get something like this. Each peak represents a different chemical compound. We can measure the relative abundance of these compounds by looking at the area under the curve. And then we divide that peak area by the peak area of an internal standard that we've added to every sample. And we added it in a known concentration so we can estimate the relative abundance of these different compounds. To reduce a lot of really hard work by a lot of people, I have this graph. Each bar represents a different species, and this is the composition of their chemical signals. The y-axis is the proportion of the total volatiles that we detected and measured in this study. On the right, the color coding represents the different functional groups that the compounds were in. The x-axis shows you the phylogenetic relationships of the species at the top. Each clade is represented by a different shape, and they're all color-coded so that no two species have the same shape and color. And the first thing that we notice is that fatty acids are a huge part of these secretions. So I've separated here saturated fatty acids on the bottom, followed by unsaturated fatty acids, because we know that physiologically they're very different. They have different functional roles, and they have different physical properties. We found a total of 47 different volatile compounds. This was a mean richness across species ranging from 8 to 30 different compounds. Between 26 and 94% of those secretions were made up of fatty acids. Another interesting thing we noticed is that aldehydes were present in a lot of these species and in really different amounts. Aldehydes are really interesting because they are fragrant. They're really odorous and potent chemical signals, and they're known to be chemical signals in a lot of other taxa. So moving forward with the phylogenetic analyses that we did, I wanted to focus on these really prominent fatty acids as well as these really odorous aldehydes. Okay, in the first part, I asked, is odor composition better predicted by habitat climate, phylogeny, or both? And to do this, we used phylogenetic comparative methods. First, I want to introduce you to our temperature variable and our precipitation variables. We chose these measures for habitat because of their known effects on the efficacy of chemical signals. We chose 2 temperature variables, including the minimum temperature of the coldest month, as well as temperature seasonality, which gives you a measure of the minimum temperature range to the maximum temperature. We also looked at annual precipitation and precipitation seasonality. These were chosen because of a previous study by Michelle Line that showed that these particular bioclimate variables are paramount in determining the type of suitable habitat for Scoloporus species. In the map, the squares are the different areas that we collected secretions from, and they're color-coded to match the different habitats. So the first thing you'll notice is these are really diverse types of habitat. They live on sandy beaches in the Yucatan. They live in tropical deciduous forests, boulders, deserts, pretty much any type of habitat you can think of, they live in. Okay, so these variables were available on the Dryad database from that study, and so we averaged these over the entire geographic range of each species. This gives us the best estimate of what the habitat was like as the species were evolving over time. Okay, here we're comparing 3 different models. Each one treats evolution slightly differently in that it says whether or not phylogenetic relatedness is important for these relationships. So the first one treats phylogenetic relatedness as unimportant, suggesting that just by knowing the phylogenetic relationship of 2 species, you cannot predict what the relationship to a particular trait is. So here we're saying phylogeny is unimportant. The second model treats evolution as very important. So it's the opposite end of the extreme. We have one where phylogenetic relatedness doesn't matter, and the other where that's all that really matters for this relationship. This is based on Felsenstein's independent contrast, and it treats evolution as a Brownian motion process, which basically says that from the beginning, you have a trait evolving stochastically and by random motion until the point of, until now. And so you can look at the phylogenetic relationship of 2 species and say they look so similar because they're closely related. The third model is an intermediate between these 2 extremes. It treats evolution as somewhat important, but not quite as important as the second model. We use the AIC value to determine the best fit model, and I like Brian O'Meara's definition of this, which says that the AIC value seeks a model that has a good fit to the truth, but few parameters. Okay, this is what we found. Species with greater proportions of unsaturated fatty acids tended to live in habitats that were colder. On the y-axis, you're looking at the proportion of unsaturated fatty acids in the secretions of the lizards. As we move from really cold, low temperatures to really warm, low temperatures, so as the habitat warms, the unsaturated fatty acids begin to decrease. We're going to look at temperature seasonality. So on the far right, this would say that there's a really big range between the low temperature and the high temperature of a habitat, so that it's a very drastic change. We're seeing that unsaturated fatty acids increase. So I think what this means is that, again, you're looking at sort of, in areas that get much colder, unsaturated fatty acids begin to be upregulated into the secretions. For this particular pattern, we found that the Brownian motion model was the best fit, suggesting that phylogeny can explain a lot of this variation and this relationship. We found no relationships to precipitation in unsaturated fatty acids, and surprisingly, we found no effect of habitat climate on saturated fatty acid proportions. This surprises me because a lot of previous literature in signaling efficacy has suggested that really stable compounds like saturated fatty acids probably help to increase the persistence of a chemical signal. So we would expect that in really hot habitats, animals would have much higher amounts of saturated fatty acids in their secretions. We found no relationship at all. We also found no effect of richness, the number of different compounds, on unsaturated fatty acids, on habitat climate. This pattern was mostly driven by 2 unsaturated fatty acids, oleic and linoleic acids. Both are common, both are oils. Oleic is odorless and linoleic is an essential fatty acid, and it's used to build many longer chain unsaturated fatty acids, so very important to an animal's physiology. We also found that species with greater proportions of aldehydes live in hotter temperatures. So again, as our low temperatures get higher and the habitat gets hotter, we get more aldehydes. Phylogeny was unimportant for this relationship, so that first model, what could explain, was the best fit. We found no significant effect of temperature seasonality on aldehyde proportions, and we found no significant effect of precipitation on aldehyde proportions. These relationships were driven by 3 main aldehydes. Octanol, decanol, and nonanol all have very strong odors. They're fruity and floral, citrusy. Particularly interesting to me was this inverse pattern we saw in unsaturated fatty acids and aldehydes. As we go from cooler temperatures to warmer temperatures, unsaturated fatty acids begin to decrease, while aldehydes begin to increase. Why this is interesting is because oleic acid we know is metabolized into nonanol, so it might explain why we see this pattern. At least some of that can be explained by metabolism itself. We also know that nonanol is attractive to mosquitoes and other blood-feeding insects, which could be very common where these lizards live. So it would be an interesting complementary strategy for foraging for them, considering they don't hunt insects. They basically sit and wait on their landmarks for something to come by and then attack it. Okay. In the second part, I'm looking at activity behavior. So we're using the same 3 phylogenetic models, but now we're asking, is odor composition better predicted by behavior, phylogeny, or both? To do this, we recorded the undisturbed behavior of lizards in the field. We recorded behavior from between 20 and 30 males, and from a distance of 2 to 20 meters. The videos were scored by many, many undergrads, 4 of which I had the pleasure of working with. Hannah Jarvis, Samantha Couch, who's here today, Clayton Harmon, and Chloe Strauss. And from these videos, we scored several different behaviors. The chemosensory behaviors tell us how chemically interested they are in general. These are baseline rates of behavior. The head bobs are bouts of head bob displays. So a series of head bobs with a pause, another series with a pause, that would be 2 bouts. Movements were defined here as long bouts of locomotion. That's at least a 10-meter transverse from A to B. And then adjustments or any other small movements of any body part. For a total measure of activity, we sum together all of these previous 4 types of behaviors. This graph shows you the summation of all this. The first thing I want to point out is that the rates of behavior on the y-axis jump from 25 scale to 100 scale after 50 because rates of activity are so high. Again, the rates of activity are the summation of all the behavior you see on the bottom plus the adjustments, which we haven't included in this graph. The first thing I want to point out is that head bobs are the highest of those 3 behaviors. They're color-coded to match the type of behavior. Chemosensory behavior is the lowest, followed by movements, and activity, of course, is much higher. So you can see that there's a lot of variation across the species, but there are some general patterns that we see species to species. The icons on the top are the same as the ones I used before. And this is what we found. Species with greater proportions of saturated fatty acids were more active. When looking at the baseline activity rates, we see saturated fatty acids go up as the activity increases. Phylogeny was important for this relationship, as the Brownian motion model was the best fit. We found this same result recapitulated in measures of chemosensory behavior as well as movement behavior. Again, phylogeny mattered for this. We found no significant relationship of saturated fatty acids to the head bob rates, which is a visual behavior, or we found no significant relationship between any behavior and unsaturated fatty acids. What I think might be happening here is that as more active species need to metabolize more saturated fatty acids to meet higher energetic demands of this high activity rate, they incidentally are diverting more toward the secretions themselves. We also found that aldehyde proportions vary with species activity and movement. So as species become more active, their aldehydes drop. Phylogeny was unimportant for this relationship, and we found no significant effect of aldehydes on chemosensory behavior or head bobs. So I want to remind you what we found with habitat temperature. Species in hotter temperatures also produce more aldehydes. And here we're showing species that move less produce more aldehydes. We know that lizards are forced to seek refuge when it's really hot out, which limits their time for other activities, including social interactions and patrolling. So this greater potency of these really fragrant aldehydes might help to boost the signals for faster detection from conspecifics, as well as potentially to detect them from greater distances. For chemical richness, we found that it decreased as activity increased. We found the same pattern for baseline head bob rates. We found no significant effect of richness, that's the number of compounds, on species movement or chemosensory rates. Phylogeny was unimportant for this relationship. So the lower energetic requirements might be allowing species to produce volatile-rich odors, and they in turn could compensate for this reduced activity by being more attractive, maybe with more numbers of compounds. We also see a trade-off between the chemical and visual signal complexity here. So as the chemical complexity gets, let me go backwards, as chemical complexity gets higher, so as richness gets higher, the number of head bobs are lower, and vice versa. So here we're seeing this sort of trade-off between a really complex odorous signal and a really complex number of head bobs. In conclusion for this third part, we showed that fatty acids are major contributors to secretions, and they change with habitat temperature and behavior. Relationships varied from a really strong phylogenetic signal to none at all. We also showed that habitat temperature drives unsaturated fatty acids, while behavior drives saturated fatty acids, but both were constrained by phylogeny. We also see more flexibility, so less phylogenetic constraint, in really fragrant signal features, that is, the proportion of different aldehydes, as well as the number of volatiles. So at this point, I want to make a case for the importance of physiology in fatty acid secretions. I think what's happening is the same physiological changes that occur in animal tissues in response to changes in habitat temperature are reflected in the chemical composition of a signal. So again, these glands are holocrine in nature, so cells burst and the contents become the secretions. So indirect selection on the physiological benefits that fatty acids provide might explain why we see so many fatty acids in secretions. We know that unsaturated fatty acids are mobilized more quickly for energy than saturated, and we found that species in hot habitats had fewer unsaturated fatty acids, so it could be that they're depleting this resource to meet higher energetic demands. Unsaturated fatty acid proportions in tissues and cell membranes also impact the cold tolerance threshold of an animal. We know that the fluidity of a membrane and its stability is impacted by the relative proportions of unsaturated to saturated fatty acids. So by altering this content of tissues, animals can stabilize physiological processes as habitat temperatures change. It's been shown that high unsaturated fatty acid diets increase the duration of torpor, lower the minimum body temperature, and metabolic rates of mammals. In lizards, it's been shown that high unsaturated fatty acid diets lower their preferred basking temperatures, but diets high in saturated fatty acids had no effect at all on their preferred temperatures. So going into this last section, with that in mind, I wanted to ask whether the variation in odor signals across a population, across one species, could tell us anything about the phenotype, whether visual or physical, of an individual, and how it might signal an individual's identity. To do this, I focused on the species Scoloporus grammicus for a few reasons. But briefly, we collected secretions from 52 males in the field, in this field site here. Why I focused on grammicus? These lizards have really interesting phenotypic differences in their throat color. So here, you can see that some males express an all blue throat color, some males have no blue at all, and then some males are in between, having some blue while also that blue being complemented by some other color, usually orange or yellow mix. Why this is interesting is because blue coloration, we know, in Scoloporus is testosterone mediated during development. So during an ontogenetic critical period, how much testosterone an individual has circulating will decide how much blue coloration they have. And after that period, the trait is fixed, so the blue coloration is there for life. Blue coloration has also been linked to higher plasma testosterone levels in adults. And we know that from a previous study, the more blue an individual has on the throat, the more aggressive it is to conspecifics. Okay, we found 31 different compounds in these 52 males. Here you're looking, the bars represent individual compounds, and on the y-axis you have the proportion of individuals that we measured that produced that compound. The first thing you'll notice is that only 4 compounds were produced by everyone. 3 of those, the first 3, are long-chain saturated fatty acids. The one on the end is a steroid. So you'll also notice that there's lots of variation in these other compounds that they produce. And so first I wanted to know, how variable are individual blends? So rather than looking at the really common compounds that everybody produces, I wanted to look at those more rare components, which are rarely looked at because they're so rare. So these were compounds that shared by less than 85% of all males. They also tended to be present in low abundances. So here, each bar represents an individual male. They're separated by throat color. Pure blue males on top, then mixed blue and no blue males. The y-axis is the percent of the secretion made up of different compounds, and they're color-coded here in this legend. So the first thing I'll say is that these are ordered. So A compounds are present in the highest abundance across all individuals, whereas V are more rare. One thing I'll point out is that while a lot of them have this A compound, a lot of them have them in very small proportions. The second thing I'll point out is that some compounds that are more rare are really highly abundant, like this one here, but other individuals don't produce any of it at all. The last thing I want to point out about how variable these individuals are is that no 2 individuals have the same mix of these highly variable compounds. Not even talking about the abundances of these compounds, just the sheer presence of the compounds. No 2 individuals have the same combination of these variable compounds. So I think these are excellent candidates for signaling individual identity in lizards. We know from a lot of behavior studies that individuals can determine familiarity, they can recognize other individuals. So I think that, that these particular compounds might be unique blends that help them recognize each other. Other studies have suggested that signals of individual identity should be highly variable and continuous in that variation, which also makes it hard to study them because if not everyone produces them, then it's hard to determine where those signals are coming from and if they're important. This concept is sort of based on a concept by Wyatt, a signature mixtures concept, who said that the information content itself is in the specific blends. So while they might not have any particular meaning, just the blend of them, the blend of the compounds themselves are the information. So similar to the way that we can recognize each other's faces. We know the face of an individual, and while it might not tell us anything about the physiology of that individual, we can recognize it and it means something to us. Okay, so in this last part I wanted to target the common compounds. We know that they could be selected for based on their consistent types of information that they can convey. If they're really common compounds, then everyone is used to smelling them and they might be able to determine information about individuals based on that. They could be sexually selected or physiologically linked. And the information content themselves is in the abundance of these signals, likely. That's what I think. Because the presence of them themselves isn't that interesting considering everybody makes them. So the abundances here are key. So for this last part I ask, do saturated fatty acids or steroids, those really common compounds, convey information about body size or throat color? We found that heavier males produce more medium-chain saturated fatty acids. So as body mass increases, so too do the proportion of medium-chain fatty acids. We found similar results for body condition. However, we found no effect on individual size, no connection to the proportion of saturated fatty acids. long chain saturated fatty acids. We also found no connection to the proportion of steroid. When we looked at throat color, so here you're looking at a non-metric multidimensional scaling plot, again using the same type of dissimilarity index as we saw for the microbes. So 2 points that are more close to one another in distance look more similarly in the chemical composition of their signals. This is color-coded, so the pure blue males have the royal blue color. And the thing that I want to point out here is that they're not grouping by color. So throat color couldn't tell us anything about the composition of these different signals. We also found no differences in mass or body condition based on throat color. However, males with blue throats produced more of this steroid, that really common steroid we saw in everybody. Pure blue males produced higher proportions than no blue males. So there's something about that blue coloration that is connected to the proportion of steroid in secretions. So we know that during development, the blue coloration is fixed, and what we might be seeing is an effect of higher T producing individuals. So if they produced more testosterone in development, perhaps throughout life they also produce more testosterone, in turn being able to divert more of a steroid to the secretions. Okay, to conclude this last part, we found individual variation in these highly variable compounds might be able to signal the individual identity that is evident from the behavioral research of other people. We also found that common compounds might be physiologically linked to other phenotypic aspects of the signaler. So a receiver might be able to tell the quality or the body size of an individual based on the proportion of medium chain saturated fatty acids in their secretions. They also might be able to estimate the aggressiveness of an individual based on their steroid proportions, since we know that really blue individuals tend to be more aggressive. In final conclusion here, I want to make 3 main points. The first is that through this thesis research, I think I've shown that territorial animals display some degree of semi-gregariousness. We see an attraction to the male secretions, and we also see that there are similar microbial communities between males and females, which could be facilitated by close social interactions. We also found that more plasticity might occur in really fragrant, smelly compounds. So as evidenced by the proportion of aldehydes and the number of different volatiles that a species can produce. Less active species in hotter temperatures produce more fragrant secretions. And we saw a trade-off in that increased rates of head bobs might compensate for less fragrant odor signals. Finally, chemical signals are mainly byproducts of metabolism. This is my main point, and the thing that kept occurring to me throughout my research. We found that fatty acid content was constrained by phylogenetic history, and that unsaturated fatty acids were driven by temperature, whereas saturated fatty acids were driven by activity level of species. We also found this relationship between testosterone and steroid abundance in the secretions, which we also know is connected to the blue coloration. And finally, we found that heavier males produce more of this medium chain saturated fatty acids. Again, tying these relationships between fatty acid, habitat temperature, behavior to physiology. Okay, and with that, I'd like to make some acknowledgments. There are lots of people to thank here. Primarily, first and foremost, I want to thank my PhD committee, Amelia, Greg, Ellen, Troy, Helena, and briefly Kurt. I also want to thank all of my collaborators that had really primary roles in all the projects I discussed today, specifically Diana Hughes, Jake Pruitt, Jaime Zuniga Vega, Milos Novotny, Irene Newton, and Joao Vital Garcia. I want to thank all of the Martins Lab members, previous and present. There have been many, and all were instrumental in helping me develop my projects, think differently about the concepts I was interested in, and really give me useful insight into what my results might mean. I want to thank many individuals that also helped various parts of this project, whether it was answering all of my burning questions or helping me do actual physical research, and all the undergraduates that helped me. All I've mentioned previously, but I also want to thank Morgan Herman, who's a student at Arizona State University. I want to acknowledge my funding sources throughout my thesis, particularly the IU President's Diversity Dissertation Fellowship, which funded me this year, as well as the Common Themes in Reproductive Diversity Training Grant. Okay, now to the fun ones. Not that those weren't fun. I want to thank my parents first and foremost. My dad has always nurtured my competitive side and my curiosity to seek out answers to the unknown. He helped foster my independence and resilience from the time I was a really young girl. My mom, whose unconditional love, loyalty, and commitment to helping me achieve my education has been a reliable constant in my otherwise crazy life. My sisters, my oldest sister Kimberly on the left, she's always been my #1 fan, like a second mom to me. She helped raise me with an unrivaled compassion for others and selflessness that is truly inspirational. My closest in age sister, Tracy, who has, from the time I was little, been my fiercest competitor. She was a very creative person, so it forced me to think on my feet and keep my wits sharp and be prepared for literally anything. She reminds me to have a sense of humor and laugh at myself, and if I don't, she'll do it for me. I also want to thank my grandparents, Manny and Carolina, that have just given me everlasting love and support and faith. I want to thank all of my friends. There's lots of them. I made a lot of great companions here at IU over the last 6 years. All of them have been really great in supporting me through quals and supporting me through my thesis writing and telling me to take a break every now and then and going on adventures with me. So all of them have been really inspiring and passionate to be around. I want to thank the love of my life, my dog Osiris. He reminds me every day to be brave and stay curious and adventurous. He's a poet at heart and a lover of the finer things, and he's always been there for the last 6 years for emotional support. And finally, last but not least, I'd like to thank my partner, Will, who has been great throughout this process. And I think what stands out the most is that I've had a tendency my whole life to be really narrowly focused and letting spontaneous whims of an idea grow rapidly and wildly into entropy. This is an issue Amelia has tried to help me with for the last 6 years. And I think Will's taught me how to pull myself out of this rabbit hole and be in a more lucid state, not necessarily one of reality, more of a fantastical realm, but he's helped me to think more broadly and thread together ideas and put my thoughts into words and think about larger concepts and help me out of situations when I've taken on way more than I can handle, so thank you. And with that, I'll take any questions. Daniel.
I have a couple. So
Sure.
Can you tell us a little bit more about who's in those OTUs that you characterize? Because it seems like asking if there are differences among sexes is one question, but at first principles you might want to know if those microbes even seem to promote the production of volatiles or some of these compounds that you think are important. So do you know who those guys are and anything about their metabolism? Like, is there reason to think that these microbes actually play a role in producing some of these compounds to begin with?
Yeah, so that's definitely a question that I've been asking myself There's a really great database called MVOC that allows you to look at the metabolism of a lot of the microbes. It's not a huge database yet, so it's hard to really pin down what these microbes might be doing. Unless you have a really specific taxa you're interested in, it's hard to know. They're very diverse. So I haven't really been able to look at specific taxa yet. There was maybe one difference in the taxa that I really trusted the sequence data for that was consistent across the databases. And that happened to be a soil-dwelling microbe, so females tended to have more, so it could just be that they're in the soil a lot more, whereas males are perched on really high conspicuous areas. So there are microbes that could be influencing volatiles. A lot of them produce a lot of alcohols and aldehydes, but I haven't been able to really pin down whether any of those microbes actually might be contributing to these volatiles.
Can I ask another question?
Sure.
So I see a lot of relationships here between certain compounds and diet or habitat or things like that. And I see a lot of hypotheses that are associated with that. In this particular taxa, are the ways that you can modify the content of or the production of some of these compounds that these animals are producing, either through dietary manipulations, like giving them a lot of saturated fatty acids, or something maybe topical? Is there any way that you can directly manipulate them to test some of these hypotheses?
Yes. Another group has done it with a different group of lizards. Specifically, they injected a bunch of different fatty acids into moth larvae and then fed them that. And by doing that, they showed that that it does influence their secretions a bit. But I'm not sure if they actually chemically analyzed them or if they did a behavioral analysis on that one. I think it was a behavioral analysis. So I don't think anyone's actually done specifically whether it influences the secretion composition. But I will say that diet probably does have a lot to do with some of the secretions that are available, but there have been other studies that looked at the evolution of diet alongside the secretion composition. Particularly, it focused on whether lizards were herbivorous or ate insects. Herbivory is more of a rare concept in lizards than insect foraging. So they didn't find any differences there based on whether they ate leaves or fruits versus insects. And there's been at least a couple other studies that have looked into diet and from an evolutionary perspective, and it doesn't seem to be super important in terms of the evolution of the secretions, but I do think that the availability of fatty acids probably has a lot to do with how many they can put into secretions. It hasn't been a study that I've directly done, but it has been one that Amelia and I, as well as Christina, have talked about trying to do with our lizards. Yes?
Yeah, so you talked a lot about like interspecific variability in some of these compounds, right? So differences between species across these pretty big climate gradients. I know you didn't do it, but can you comment on what you might, like for a lizard that has a fairly broad geographic range, if it would show a similar pattern? So just within that one species, would it have, do you think it would be connected to climate in the same way, or?
That's a good question. So there definitely are species we could do that with. The one here at Lake Monroe is hugely widespread.
In Tennessee, right?
Undulatus, yeah. So we also took them from Tennessee. They can be slightly different subspecies. There's taxa, phylogenetic relationships are constantly up in the air and being debated. But yeah, I think where they are probably influences the secretions that they produce. Particularly because those habitats are so different. Not only temperature, but like I showed, it really influences a lot of their social dynamics and their activity periods. So I think all of those interactions are important, and I might be able to predict based on my data how it would influence them. But I also think that it's really important to consider things like population density, how close they are to one another, and how far their signals have to travel or how long they have to last. So I think that by modifying behavior, lizards can sort of compensate for a lot of these phylogenetic constraints on physiology. So yeah, whether or not the undulatus here look anything like the undulatus in Tennessee, I imagine that they are, because they're probably also behaviorally different, I imagine that their secretions are slightly different. I think that they'd still have really high proportions of fatty acids and have all the same functional groups that I showed today, just judging by those 12 species. Even though they're really diverse, they tend to produce really similar types of compounds. It's just in the specific compounds that they're producing and the abundances that change. Jake?
Please help my memory. You found a relationship between the number of compounds and the number of head bobs. Was it per minute or per display?
These are not counts per display, these are bouts.
Okay.
So,
So
yeah.
lizards that displayed more frequently in the environment had more compounds in their secretions. Is
The opposite.
the opposite.
It was an inverse
Okay.
relationship.
Yeah, like I said, my memory needs help. What about color? Have you analyzed the number of colors a lizard has, where the color is distributed, if it's all over the body, if it's just in one discrete patch?
When you say color, do you mean ventral color or throat color?
Well, all of the above. So you could have, a lizard that just has one blue patch on its belly, or a lizard that has blue on throat and belly, or a lizard that has 4 colors distributed across its body.
I haven't for this study. I did try to look at the number of throat colors in the individual variation study. Didn't seem to matter. But in terms of evolution across species, it'd be interesting to look at. It'd be hard, unless you're just doing a really crude count of number of colors. You could take spectrophotometer data and look at the curves. I don't like to do that stuff. Christina might. But if you did a really crude count, you might be able to. I mean, species like Mariami have a rainbow on their belly, so it really depends how you're defining colors there. But yeah, I'd have to look back at these species and see what they are to see if maybe the pattern is consistent.
Okay, let's thank her again. And we're going to have to ask everybody, as a traditional defense, everybody to leave now. And actually, Stephanie, you have to go out.